Hey guys, welcome back. So today we're going to be looking at the 3-point Testcross. Last time we looked at the 2-point Testcross. So just to refresh your memory, looked at the 2-point Testcross. What we were looking at here was going to be 2 points on a chromosome. So we compared that to 2 points on a physical map of Hyrule. So the 3-point Testcross in relation to that is exactly what it sounds like it would be. So it'll be a third point somewhere between these two. Okay, so we have genes A, C, and B, and again, 3 points on that map of Hyrule. So a real example of this, we think about 3 genes corresponding to 3 physical traits. We can think about a plant that's going to have 3 genes for fruit morphology. So these 3 genes, these 3 traits, are going to correspond to the color of the fruit that's going to be produced, so either yellow or purple, so that's one of our traits. The overall shape of this fruit, so elongated, sort of a longer shape, or round, and we're going to also see sort of the juiciness of it. Is it going to be kind of dry, or is it going to have more juice? Okay, so these are 3 traits. They're going to correspond to 3 genes, and we know that they're in there somewhere. We've done our homework. We know that they're going to be on the same chromosome. We've already done all that chi-square stuff to make sure that we're going to be dealing with traits that are linked. But what we don't know, is it shape, then color, then juice? Is it color, juice, shape? Is it going to be juice, shape, color? So that's the question that we're going to be answering. In addition, we're going to be finding that relative distance, that recombination frequency between these genes. So let's start out with how we actually set up this 3-point Testcross. Okay, so the word Testcross is in there, so that means there's going to be a cross. So it's important to take into account the actual genotypes of these parents that we're about to use. So one is a triple heterozygote, which means that it is heterozygous for all 3 of these genes, all 3 of these traits. Now the phenotype that it's displaying is what we would consider to be wild type, or the dominant phenotype in this case, but this is going to be heterozygous for all 3 of these traits. The Testcross, kind of the hallmark of that, is crossing this to an individual that will be homozygous recessive for all 3 traits. So this one is going to be recessive for all 3 of them. So we set up this cross, okay? So these are plants. We're going to let pollen do as it does. We have now these 2 parents, this wild type heterozygous individual that looks yellow, elongated, and dry, crossed to this homozygous recessive individual that looks purple, round, and juicy. We let nature take its course, and then as offspring are produced, what we're going to do is count the different phenotypes that we get. What color is it? What shape is it? And what is the juice content? So of course, we're going to organize this into a beautiful set of data right here. So we've got some numbers. We look at the phenotypes. We have yellow elongated dry, purple round dry, all kinds of combinations, purple elongated juicy, just all kinds of combinations that come out of this. And we've recorded the actual number of offspring that have these physical traits. And so remember from the last video, we emphasized that we're going to be using the physical appearance of these traits, the phenotype. That's going to help represent what's happening at the chromosomal level. So we're going to be using those phenotypes to help inform us as to what's going on the chromosome. So in order to determine the order of these genes, it's probably best to start categorizing these offspring. So we're basically going to start categorizing them into a parental phenotype and recombinant phenotype. Okay, so take a second, think about this, and think, can you organize these into different phenotypes? Starting off, can you find the parental phenotype? Okay, welcome back. So now we're going to go ahead and make sure that we're on the right track. So hopefully this was not too hard for you. Remembering that the parental phenotype is going to be in the offspring what exactly matches the parents. So if we had a yellow, elongated, dry parent, then one of these is going to be a yellow, elongated, dry individual. The other parent, purple, round, juicy, that's exactly what we're going to be looking for as a parental type of offspring. Okay, so we can see those are going to be our parental phenotypes. Another good hint when you're looking at data like this is realizing you've got two parents, so you're going to have two categories that are going to be parental, and the numbers are often going to be a fair bit larger than the rest of these. Okay, that's because recombination doesn't occur where we want it to 100% of the time. Okay, it's all about probabilities. So usually we're going to see these parental categories being quite a bit larger. So that can kind of help like tune you in to maybe like double checking these phenotypes. But in its most basic sense, we're literally just matching the phenotypes. Okay, so now that we found the parentals, it's important to note that in our 3-point test cross, all of the rest of these are going to be recombinant. Okay, so they're all going to be recombinant, but we can't just stop here like we did with the 2-point test cross. Okay, we have a little bit more work to do because we've got 3 genes involved. So in order to understand how to effectively categorize these offspring into recombinants that are going to be useful to us, we have to consider the way that recombination could have happened in this cross. So what are the possibilities for useful recombination with respect to these 3 traits? So let's look at these 3 chromosomes. Okay, again, we've got example genes A, C, and B. Okay, so again, 3 ways this can work. The first is that we can have one crossover occur somewhere between A and C. Okay, somewhere between A and C. That's one possibility, with the rest of this being left alone. Okay, so now we can think about a second possibility as being a single crossover between C and B. Okay, so we've just mixed and matched here. Again, leaving this alone between A and C at this time. The third possibility is what we call a double crossover, which exactly as the name suggests, there will be 2 crossover events, one between A and C, and a second one between C and B. Okay, so last time in the 2-point test cross, we didn't have to worry about these double crossovers, but we do this time because it's moving around the gene that we're interested in. Okay, if there was a double crossover anywhere else, we don't care. It's because it is involving the gene that we're interested in. Okay, so those will be important to note. Okay, and one thing that is going to help us out is to realize that double crossovers, okay, we did have a crossover between A and C, and then we had a second one between our C and B right here. That's going to be important in a little bit. And another thing that's going to be really helpful is to remember the association between what we see as the phenotype and what has happened on the chromosomes. So in this case, we've seen that this gene has been exchanged, mixed and matched. That means the phenotype will be the one that is decoupled. So in the parent, we had, to begin with, on this darker blue chromosome, previously recessive A, recessive C, recessive B. A and B are still recessive here. They're still in coupling. It's gonna be the C that's now got the wild type version. This is gonna be decoupled. So we're gonna be finding the double crossovers first. That's gonna be our first step, okay? But in all of these, the phenotype is gonna tell you which gene is decoupled. So for example, whatever B is, that's the one that's decoupled in this example. In this first example, we're going to see that A is the one that is decoupled, okay? recessive C and B still together, wild type C and B still together. The one that is decoupled tells you kind of where this happened. Okay, so I mentioned that the first thing that we're going to do is look for the double crossovers. There's going to be a reason for that. Okay, so we'll go ahead and look at these double crossovers. Okay, and so the reason we're going to start with these guys is because they're going to help us understand. We've got juice, color, shape. Okay, whatever phenotype is going to be in the middle, we can exchange it and we will actually see the phenotype change. Okay, so I'm not saying that color is going to be in the middle, but that's an example. That's the easiest one to show on this animation on PowerPoint. We're going to actually, again, if we don't have the crossover, we won't see it, but once we do, we'll actually see the phenotype change. And so the double crossover is going to be important because it's going to tell us which gene is in the middle. Okay, so we're actually going to be able to figure that out. We can also pretty easily pinpoint these based off of the offspring frequencies. So let's revisit these offspring, remembering that a double crossover means there were two crossovers, okay? And the essence of this is basically probability or chance. What is the chance that one crossover will happen in a region? That's going to be more than the chance of two happening in that same region. So having two of them happen in the same region is less likely. That will produce fewer offspring. So when we look at this table, go back to the actual counts. Remember, we are interested in the double crossover because it's about to tell us what gene is in the middle. Okay, but they're easy to pinpoint because it is less likely to have two recombination events in that region. Pretty easy to pick out these low numbers. Also, again, keeping in mind that we're working with these phenotypic categories in pairs. That's because we've done a mix and match, so both of these are involved, okay? This lighter blue chromosome's involved, and this darker in whatever offspring they end up in. So we're working in pairs. These are going to be our double crossovers, okay? So take a look at this for a second. Remember that the phenotype is going to help us understand which gene is decoupled. We know that the double crossovers are going to tell us which gene is in the middle. From looking at these phenotypes, do you know which gene is in the middle? Okay, welcome back. So hopefully what you've seen here in our double crossover category are parentals. If you remember, we've got them here. We had one that was yellow, elongated, and dry. A parental that was purple, brown, and juicy. So in these double crossovers, we're looking for the one out of three of these traits that's going to be different from the parental. So if yellow, elongated, and dry is one parental, then yellow, elongated, juicy is the one that's decoupled. We can see that up here as well. The original parental phenotype being purple, brown, and juicy. Here, purple and round are still together. Okay, so that's going to be like that A and B still together, but now dry has been exchanged. So we know that juice is in the middle. Okay, that's going to give us a really good jumping off point because the next couple of phases of this, we're going to end up calculating the distance between whatever gene's over here and juice and whatever gene is over here and juice again. So we're looking at these two regions. We're going to divide these out into two regions. We've dealt with the double crossovers. So both of the rest of these, like all of the rest of these are going to be single crossover events. So just one crossover, either here or here, but not both. Okay, so we can start by dividing the area into regions. Okay, I'm going to call this to the left region 1. for right now, it's not going to really matter what gene ends up being over here or over here. We're still going to get the correct distance because we're going to use the numbers in the table. Okay, I'll use the other one as region 2. We're going to calculate them separately and then kind of put it all together relative to where juice is. Okay, so we've got our regions divided. We're going to be working in region 1 first. Okay, so what we need to do is actually kind of group the remaining single crossover categories together, remembering that these numbers are generally going to look paired. So we saw in the double crossover category, kind of similar numbers here, okay, similar parental numbers. So in order to pick out whichever region we're working in, okay, we want the numbers to match. So 22 and 22 are going to match here. 52 and 46 are going to match here. Okay, I'm going to start with the 52 and 46 and these two categories, okay, to work in region 1. And we have to remember, again, the phenotype will tell us which gene is going to be the one that is decoupled. What is the trait that's decoupled here? Okay, so I'm going to end up looking at this. And if we put these together, we have purple, elongated, and dry. Okay, and we compare that to our other category, yellow, round, and juicy. Okay, so we can take a look back at our parents. Yellow, elongated, dry. We can see that elongated and dry is still together. We had purple, round, and juicy. For some reason, I feel it helps if I say it really fast, like one word. It kind of helps me think about what goes together. Okay, round and juicy. And we can see that the color is going to be what is decoupled. Okay, so purple and yellow are what are going to be exchanged. So in our region 1, this is color. Again, if you had started with this other category and called that region 1, these two remaining ones, that would be fine. You're still going to get the same distance. Okay, you'll still get the same distance between them. And it's okay, the chromosomes align. It can flip back and forth. It can be upside down. Okay, as long as you calculate the correct distance, pick the two matching categories. That's what we're going to work with. Figure out which of these traits is decoupled. That's the gene you're working in. So our region 1, because I've chosen these two, is going to be the distance between color for this color gene and juice. So let's get some math going here. We're going to do a math here. Okay, so we can kind of get started. Again, this is the region we're working in. We'll be working in region 1, okay, color and juice. And here's our formula, and this looks like a really bad formula. This is a percentage. It's still recombination frequencies, but because we've had 3 genes with this potential for double crossovers to come into play, we just have to add a little bit to it. It's still a percentage. It's still a recombination frequency. So here's our formula. Let's break it down. So this is just the recombination frequency in region 1, which I've just defined. That's going to be the number of recombinants that happen in region 1, which we're going to have up here. I've just reminded us all A to C means is that we're going between A and C here. This is not math. This is just to let us know where we're working, region 1, so the number of single crossovers in that region, which again we've already highlighted, ready to work with, plus the number of double crossovers. Okay, so the double crossovers highlighted in green. The reason for that is going to be because this is a double crossover. Just because 2 happen doesn't negate the fact that one did happen right here. Just because another one happened here, this one that happened is still important. Okay, the one that happened between A and C still counts. Okay, so there were 2 of them. All right, this, again, multiplied by 100, this is just going to be a percentage. It's over the total progeny. Okay, so we can see how that's going to work out. All right, you can kind of see these crossover events. Again, we're working here. And with these double crossovers, we do have to account for this double crossover. So let's get back to the actual formula. Okay, and so what you'll do is take a second, pause that video, and see if you can figure out what that recombination frequency or distance is going to be in region 1. Okay, welcome back. So let's map this out. It's not going to be too bad. So if you remember our formula, we had, again, the number of single crossovers plus the number of double crossovers in that region. Here were our single crossovers. Okay, we categorize them together because they have similar numbers, and then use the phenotype to see which gene's going to be decoupled. We decided it was color. Okay, remembering we have to add in those double crossovers because, again, a crossover event did occur in this region. Okay, so we add these together. Okay, so this is just math. Our 46, 52, 4, and 2, all added together, the total number. So just add all these guys up. We end up basically with 104 over 500. Multiply this to get a percentage, 20.8%. That's how many recombinants, that's the percentage of recombinants, which translates to a genetic distance in region 1 of 20.8 centimorgans. So now we know the distance between color and juice, 20.8 centimorgans. We can get ready to put the rest of that map together. Okay, so I think it's pretty clear that the next step we're going to take is to be calculating this distance between juice and shape. Okay, so let's get started with region 2. Remember this formula. This is the recombination frequency in region 2 is going to be equal to the single crossovers in region 2, which I haven't highlighted because by default they're left there, but we could pick these out because they have identical numbers, very similar numbers. Okay, single crossovers in region 2, and remember this is just notation. Region 2 is between C and B here. Okay, so this is no subtraction, these aren't variables you need to solve for. Okay, just any crossovers that happen between C and B, whether they be single, which we've accounted for here, or double. Remember, there's our crossover, and then we have to account for that crossover. So yes, the double crossovers get counted twice, but that is because they had 2 crossovers. Okay, so it's going to be that same percentage. Okay, so again, working in region 2, we've had an example for region 1 now. See if you can figure out the distance between C and B, or that's going to be our juice and shape. All right, so let's do that math. Okay, so you can see again our single crossover events happening between C and B, okay, which we just know what's left. But again, you can still see that the decoupling occurred here. The round and elongated are going to be what is going to be decoupled, so that's where we're working. Numbers are the same. Those are our single crossovers. Again, adding in those double crossovers, which you've accounted for. So again, our 22 plus 22 plus 4 plus 2, we end up with 50 over 500. Okay, and again, this is just a percentage. This is just the total, added all these guys up, times 100, we get 10%, again, translating to 10 centimorgans. So we've actually started mapping these traits where you can see the distance between them, remembering that in general, a larger genetic distance, a larger recombination frequency, centimorgan, map unit, that is loosely going to translate, there's a relationship between the actual physical distance when we think about nucleotide sequence. Okay, so we don't exactly know what the sequence is, remembering this is an indirect way to measure it, but the idea is that there's a larger chance for recombination to happen in longer regions than in shorter ones. So these roughly correspond to the actual physical distance. So now we know the basis of genetic mapping. You can kind of conceptually at least expand this out into the entire genome. There are going to be some things that are a little bit different when you're mapping in the entire genome. There won't always be a really clear relationship between physical distance and this recombination frequency. There are simply some areas in the chromosome that have a harder time undergoing recombination. Okay, these are going to be things that are more tightly locked down, things that are going to be more heterochromatin or more tightly packed. Areas around the centromere and telomere, okay, those are going to be a little bit more difficult. But generally, once you take a lot of markers together, which is what we're going to be looking at in the next video, you can start to get a good idea of what these genetic distances, these map units, are going to be along all of the chromosomes, along the entire chromosome. Okay, so in conclusion, we hopefully understand now how to use what we observe in the phenotype to find recombination frequencies, and we've learned to translate that into relative distance in the form of either map units or centimorgans, whichever term you want to use. So in our next video, we're going to be looking at mapping in the whole genome. So we won't be doing more of these recombination frequencies necessarily, but what we're going to be looking at is finding those destination genes like Hyrule Castle that I introduced to you in the first video. So I'll go ahead and end today with our biology joke of the day.